the South Florida Arboricultural and Landscape Professional webinar series. We're very lucky to have Dr. Bahder here in South Florida. He's worked a lot with palms and entomology specific to palms. And I mean, he's been a huge resource for lethal bronzing disease and is probably the foremost expert of it in the definitely in Florida, maybe the world. So with that, I'm going to go ahead and pass it off to Dr. Bahder here.
Thank you, John. All right. Can everybody hear me okay? My audio is still coming through. Let's see.
I'm hearing you clearly.
Okay, so I'll just assume everybody else can. Says yes, audio, video good. Okay. All right, so I guess we can get started. I'd like to thank everybody for tuning in to today's webinar, and especially a big thanks to John for inviting me and making it happen. As he mentioned, I'm specializing in research of lethal bronzing in palms. It's primarily a Florida issue, but it is in Texas and at least Louisiana, maybe in some of the other Gulf states as well. But it's also in a few other countries around the Caribbean basin. But it appears to be most damaging here in Florida. And I joined UF in 2016 as an entomologist to look into what insect was transmitting it, but then also to try to develop management programs for the vector and the disease as well. So today I just want to share basically where the research program is at, some new findings, and some, I guess, hopeful messages for future management of the disease. And before I get into it, just a quick thanks to all my funding agencies and students and staff who've helped and really been instrumental in getting the data and basically my boots on the ground to make this research happen. And with that, I'll give a kind of a brief background on phytoplasma diseases in general in the Caribbean, and then kind of connect that to where our current problem with lethal bronzing. So phytoplasma diseases in the Caribbean have been around since the late 1800s. LY, which some of you may be aware of, was the primary disease in Florida in the 70s and 80s that primarily hit coconut palms in South Florida. This disease was first documented in Jamaica in the late 1800s. Then it showed up in the Florida Keys in the early 1930s and caused a lot of economic loss to the landscape companies because it killed off so many Jamaica tall coconut palms in South Florida. But then it kind of petered out a little bit into the late 90s and early 2000s, and it's still currently around, but it's not nearly as prevalent as lethal bronzing currently is. And here I just wanted to show a map of the confirmed cases of palm phytoplasmas, whether that's lethal yellowing or lethal bronzing in the Caribbean basin. You can see it goes as far south as Honduras in Central America and in Guadeloupe in the Caribbean islands. And then naturally it's up into Texas. That's not part of this map, but this kind of gives you an overall look at the distribution of the phytoplasmas diseases in this region. And like I said, LY historically has been the major disease and is still pretty prevalent in Jamaica and a lot of the other regions or other countries in the Caribbean. But in Florida, lethal bronzing is kind of displaced LY and taken over. Now, I mentioned that LY was introduced in the Keys and then spread north to about the southern third of the state of Florida. LB has a little bit different history. It was introduced sometime around 2006, maybe before that, but it's been here for the better part of a decade and a half, and basically it was introduced around the Tampa area and has since spread throughout the entire state. So you can find it as far north as Jacksonville on the northern limit in Florida, and then the southern limit in the Florida Keys in Monroe County. And then as far west, we have cases in Pensacola. Now, while the current county distribution map, we don't have records from every county. At this point, if you look, you could probably find it in just about every county. It's everywhere. And on this map, you can even see the cases that are shown from Louisiana and Texas. So I don't doubt that it could be in Mississippi, Alabama, and Georgia as well. Now, while it's present throughout Florida, it's still more concentrated in the central part of the state. So if you go from Tampa southeast, kind of diagonally to about West Palm Beach, that's where you see the majority of the infections. We have it in Broward County and Miami-Dade and Monroe, as I mentioned, but it's not nearly as prevalent as it is in Central Florida. And in North Florida, it's starting, from what I've heard, is starting to increase in incidence in Gainesville, as well as Jacksonville. The number of documented cases we're getting from the clinic samples is significantly increasing in those areas. So it appears that the disease is still picking up speed. As it gets more established in South Florida, I do expect to see more and more an increase in disease incident just because of the density of palms as well as the diversity. So over time, LB has spread not just in space, but it's continually jumping into new hosts. And currently, we are at 21 confirmed host species for lethal bronzing. This table here summarizes the host range of all of the phytoplasmas in the Caribbean basin. So the column that says subgroup is corresponding to which phytoplasma we're talking about. Subgroup A refers to lethal yellowing. Subgroups B and D are the lethal bronzing phytoplasma, and E is kind of an obscure strain that's only known from Mexico and the Dominican Republic. So this kind of shows the scientific name, common name of a lot of the ornamental palms that are affected by lethal bronzing here in Florida. And these images here show some of the newer host records we have for lethal bronzing. And like I said, the spread south so far has been faster than going north. And I attribute this just, as I mentioned, to the higher density of palms and the higher diversity. And going north, pretty much north of Orlando, you kind of stop seeing large stretches of palms. You get clusters of cabbage palm in the wild along the roadways, and then some palms in the urban areas, but they're not nearly as prevalent as when you're further south. So it is spreading northward, but at a slower rate than it is going south. So for symptoms, I'm sure everybody's at least somewhat familiar with what lethal bronzing looks like. And one of the challenges with symptoms is it is basically a physiological response to the phytoplasma inundating the vascular network. So as the phytoplasma moves throughout the plant and blocks up the phloem or the sap, basically it's starving the plant to death, and it manifests in a manner that's very similar to other stresses that cabbage palm can face, such as burn damage or transplant stress. So the symptoms, if you're watching the same plant over a long period of time, you can use those symptoms as a diagnosis, but generally confirming with a molecular test is the best way to make sure that the causal agent of the symptoms is lethal bronzing. And basically the symptoms here in this picture, they start with a premature fruit drop and a necrosis of the inflorescence. And in all phytoplasma diseases of palms, whether it's LY, LB, or the subgroup E, we call it lethal E, basically the first signs of the phytoplasma infection are going to be the premature fruit drop or the inflorescence necrosis. Now, you won't always see this in a stressed plant, or if it's a fungal infection like Ganoderma, the palm will still maintain the fruit or inflorescence. So that's always going to happen in the phytoplasma infections. Now, that's assuming you're at a time of year when the plant's putting out flowers or has fruit on the inflorescence. Now, in a species like coconut, where pretty much there's always fruit or flowers emerging, it's fine. But in some species like the phoenix palms, where it's a little more temporal and they only put out one fruit cluster a year, if it's at a time of year when it's not fruiting or flowering, then you're going to miss this symptom. But following that initial symptom of the fruit dropping or the inflorescence necrosis, you're going to then start seeing canopy loss, where the oldest fronds in the canopy, which are the ones lowest to the ground, start to desiccate and dry up. And over time, that canopy loss starts affecting younger and younger leaves. So you kind of get this layered effect of dead fronds on the palm that produce, in general, this bronze coloration that's distinct from naturally senescing palm fronds. And this canopy die-off kind of layers and marches further and further into the canopy. Then approximately at 50% canopy loss, you see the spear leaf collapse due to the death of the apical meristem. And at that point, the palm is completely dead. Now, the timeline for this symptom progression is quite variable. It can be as quick as under a month from symptom onset to complete death, but I've seen some palms linger for up to 8 or 9 months. So there's variability in how long this symptom progression goes, but I'd say on average you're looking at 3 months from symptom onset to the palm's death. And as far as we know, it's always lethal. So any palm infected or testing positive for lethal bronzing is going to die of the infection. We have not found any palms that are resistant and can maintain the phytoplasma, but continue growing. Okay, so now I'm going to kind of switch gears and talk about the insect vector of the disease. LY and LB are both similar. They're very closely related pathogens. They cause this functionally they do the same thing. They kill any palm that they infect. And as it turns out, the vector for both lethal yellowing and lethal bronzing is the same insect, Hoplaxius crudus. You can see here is an image on the right-hand side. You have the male. It's a little bit smaller, paler in color. And on the left, you have the female with a little bit more robust body and a little bit darker coloration. And this insect was confirmed as the vector of LY in the late 70s. And just recently, we determined that it was capable of transmitting the phytoplasma, the lethal bronzing phytoplasma. So when we were doing our research on to confirm the vector, basically we were exposing insects to spear leaf material from infected palms, and basically allowed them to feed for up to 5 days on these palm fronds. And then we would do two different things. We would transfer them to an artificial feeding, an artificial diet. Basically, it was sucrose, 10% sugar and water, and we allow them to feed over this parafilm membrane. And then we would take the sucrose out of the container and test it by digital PCR to see if we could detect the phytoplasma. And here you can see these colored squares where they have both yellow and blue pixels. The yellow pixels represent negative reactions, and the blue are positive detection of the phytoplasma. So the amount's very low, but it is present in the feeding media. So this confirmed that the insect could transmit the pathogen. And then we also did some experiments where after they fed on the palm frond, we would dissect the head and take out the salivary glands, and we were able to detect the phytoplasma in the salivary glands. And any insect that can basically acquire the pathogen in the salivary glands is going to be a vector of that pathogen. And we found that the insect needed a minimum of 2 days of access to this plant tissue to acquire the phytoplasma in the salivary glands, and 5 days is optimal. And this is kind of really important information that I want you to remember for the upcoming slide when we talk about management of this insect. But with these experiments, we were able to show that it needed this amount of time to acquire the pathogen. And the next stage is to do experiments where we transmit the pathogen to a plant itself to establish the true latent period of the phytoplasma. We do have some experiments we've conducted with Christmas palms and Prochardia palms where we have gotten a few palm seedlings to die, but we're currently in the process of analyzing that data. But it's looking like the latent period is approximately 4 to 5 months, which is consistent with what we've seen in some field settings where there's spread of the disease. Okay, so this will be one of the first slides on vector management and some data we found from doing some insecticide trials with Phoenix sylvestris seedlings. So what we did was we treated these palm seedlings with different formulations of imidacloprid, and we used formulations that were registered and are approved for use in turf grass. And I'll explain why we use that in a moment. But basically, from this, you can see that we tried a soil drench, a granular, and a spray treatment for imidacloprid on these palm seedlings. And you can see the control is the red line, and the granular did not have a really good treatment effect. You're still getting, live insects, not many, but at almost 2 weeks after the insects were exposed to the plant. But the spray treatment and the soil drench gave us really good mortality. So you can see within 24 hours of the insects exposed to these plants, almost 90% of them were dead within the first 24 hours. I mean, even within the first 6 hours, you can see there's a difference in mortality. But once you get to 24 hours, that's where you see a huge treatment effect of both the soil drench and the spray treatment. And then you can see at day 2, you have approximately 95% mortality. And then at day 4, for the soil drench, you have 100% mortality, and basically the spray treatment is about 99% mortality. Now, if you remember, I said that the acquisition of the phytoplasma is optimal at 5 days. It needs a minimum of 2 days, but basically the majority of the insects are dying from their exposure to imidacloprid before they have time to acquire the pathogen. And in a lot of these systems, what happens is the inoculation time is comparable to the acquisition time. So when we did our experiments on the plants, we gave them 5 days access to these plants to infect them, and basically found that that's what was needed to infect the recipient plants. So what this tells us is that the insecticides are killing, specifically imidacloprid in this case, the insecticide is killing the vector faster than it can acquire or transmit the phytoplasma. And this is important because basically it indirectly can help protect plants from infection. Now, up till this point, we've been relying on antibiotics, oxytetracycline, as a preventative treatment in palms to protect it from phytoplasma infection. And in that case, the antibiotic actually has action against the phytoplasma itself and protects the plant. But it's not perfect. It doesn't work 100% of the time. And it's hard, it doesn't get fully systemic in the plant. So if it doesn't cure the plant, it can only be used as a preventative. And if it's not done properly, it does fail on a somewhat frequent timeline. So with the insecticide, it can indirectly protect the plant. Not that it's having an effect against the phytoplasma itself, but it's killing the insect vector faster than it can acquire or transmit the disease. So using the insecticide is good because you're simultaneously protecting the plant, but also reducing the vector population locally. So, I'm not saying to not use the antibiotic, but I've seen it fail on a somewhat frequent basis. So having this additional tool to help manage the vector, but also protect the plants, I think is going to be important, especially in urban settings. And I make this distinction because in the nurseries, the in-ground nurseries where the palms are grown, basically, around 95% of the population of nymphs live in the ditches between the palm beds. And the nymphs of Hoplaxius crudus don't actually develop in the palm canopy. They feed on at the base of grasses. At this point, they're documented on around 40 some different species of grass between here in Florida and some of the research out of Mexico. So basically with the insects primarily inhabiting the grass in these ditches, this is a very clear, defined, and easy target for management. I've visited nurseries where they basically clear cut the grass in the ditches once a year, and that pretty much eliminates the crudus populations from the palm plots. So in palm nurseries, management of Haplaxius crudus is very simple. You can use cultural practices such as rototilling the grass in the rototilling the ditches between the palm beds once a year, and that's going to keep your vector population out of there. Alternatively, you could treat with these grasses with insecticides if you want, but it's not necessary. Just removing the grass is going to be perfectly adequate. Now, this approach is not possible in urban settings because you have you don't have big ditches between all the different palms that are out there. It's, you know, you have people's yards, medians, green spaces, parks. There's a lot of different habitat types. So nailing down the distribution of the nymphs in the urban setting is much, much more difficult. And this gets into why we were using the turf grass formulated imidacloprid in our trials, even though we were testing them on palms, is because in urban settings, the grass on the medians or in people's yards can be treated, and oftentimes are already being treated with imidacloprid to control things like ground pearls and chinch bugs. So it's a cheap and easy tool that's already available that's going to have an effect on the Haplaxius crudus nymphs. Now, we also found that in the nurseries, there's seems to be a preference in terms of what size palms the adults are breeding on. And it seems that the palms that are shorter tend to harbor, in those plots where the palms are smaller, those harbor more nymphs, significantly more than taller plants. And again, this provides a useful target for nurseries to manage the vector population. But in urban settings as well, it gives some clues as to where the nymphs may be more prevalent along highways or in parks, to kind of look for palms that are shorter, where the fronds are closer to the grass, and it makes it easier for the adults of Haplaxius crudus to get to those breeding habitats. So targeting grasses in urban areas where you have kind of shorter palms, in areas where there's like depressions at the base of an overpass, at the bottom of that hill where you get kind of a dip and water accumulates. Those are going to be likely places where the nymphs are going to be more common. I'd like to do some experiments to look at the distribution in these areas, but it hasn't worked out yet. But with how cheap and easy it is to apply imidacloprid on turf grass, you can do larger areas just to make sure you get good coverage and hit all of the potential habitat for these nymphs. And also, that's going to help lower the vector population because you're targeting the nymphs in the grass. Now, you can still treat the palms themselves with imidacloprid and insecticide to target the adult stage as well. And then you get the added benefit of potentially protecting the plant from new infections. So now I want to talk about some new data that we have that was just recently published, and it's kind of an exciting area that I'm hoping to be able to continue with, is looking at volatiles emitted from lethal bronzing infected palms. So a few years ago, we started realizing that there were more insects as well, excuse me, the vector was more prevalent on lethal bronzing infected plants rather than healthy plants. So we were curious that there might be some kind of chemical cue that the insect is detecting and orienting to, and we were hoping to identify these compounds and maybe use them in a management context for, developing an attract and kill technology, or potentially a repellent type spray for the plants. But when we started looking at this, we found some interesting data when we characterized the volatile profiles from infected palms and then nearby palms that were healthy, but were kind of either immediately adjacent or in the general vicinity of lethal bronzing infected plants. And what we found was that the infected palms, you can see on this graph on the left here, that cluster of bars shows a distinct chemical signal that's being emitted by lethal bronzing infected plants. So the blue bar represents hexanol and the green one represents 2-hexanol. And these two compounds were in elevated concentration from lethal bronzing infected plants. The threatened plants, which were healthy, that were nearby, did not have those elevated concentrations of those two compounds, but instead were producing a specific compound that was unique to only the plants that were threatened, or that felt threatened, quote unquote, by being close to the infected plants. That 3-hexanol is a unique response that it was producing as a result of the increased concentration of the other two compounds from infected plants. So this we found was interesting. It indicated that the infected plants were emitting a signal and plants nearby were responding in kind by trying to defend themselves. And we found that this compound that's unique to the threatened plant has antimicrobial properties, and it's often used in post-harvest production of a lot of fruits, where they treat the fruits with this compound to prevent fungal or bacterial growth on the fruit itself. So this told us that the plants were trying to protect themselves with their own innate, not a true immune response, but they were producing something that would hopefully protect the plant from the infection. And we noticed that in some stands of palms, you'll get a couple of infections, but nearby plants will be perfectly healthy and will stay fine for years. But in that time frame, you'll see palms that are further away start to get infected. So what we think is that this response that these plants are getting to being near infected plants is enough to some extent to prevent infection. But this is where kind of we had to stop with the study. We need to get some support to try and test these compounds to see if they are effective at both preventing infections as well as maybe curing the infection. So the idea with this going forward is to take these alarm compounds, the hexanol and 2-hexanol, which are emitted from the infected plants, and basically putting dispensers out and trying to trick the plants into producing their own immunity, and that may help and prevent transmission, or excuse me, infection of lethal bronzing. So this would be a very cost-effective way to kind of give immunity to a lot of palms over a large area, and you wouldn't have to inject or drill holes into the plants that you're treating. Then the other idea would be to take this 3-hexanol that is produced by the threatened plant that appears to be effective at protecting the palms from LB and injecting this into palms that are kind of early in the disease cycle. And if we can get it early enough and give them high enough dose, we might be able to reverse and potentially cure the infection. So those are two areas of research we're interested in pursuing in the coming years to develop a new management tool for lethal bronzing. We've done some preliminary experiments, and basically we injected the 2-hexanol, the alarm pheromone, into not pheromone, excuse me, the alarm volatile that the infected plants produce, and we injected that into healthy palms, and we found that that caused the plants to start producing 3-hexanol. So we know that that compound elicits that response in a healthy plant. And when we injected 2-hexanol into LB infected plants, we noticed a distinct drop in phytoplasma concentration approximately 3 weeks post inoculation. We weren't able to continue this because the funding for that project ended and the postdoc doing the research left, but it's preliminary results that we're excited with, and we want to gives us confidence to continue pursuing this as a potential management tool in the near future. Okay, so now I want to talk a little bit about where we are with diagnostics, because a lot of you send in samples for us to confirm the infection status of a plant, and we're continually trying to improve our technology and our approach to doing this, where we can detect it faster with greater sensitivity and with greater accuracy. And just recently, we developed a new test based on a different gene that allows us to amplify and distinguish between all 3 of the phytoplasma I mentioned. Now, I know the focus of the talk was lethal bronzing and a little bit on lethal yellowing, but we also have this lethal E included in this test, and it basically allows us to, in the same reaction, detect and distinguish all 3 of these phytoplasmas. Now, there appears to be some difference in host range between LB and LY, and we don't know what the host range of LE is. So being able to distinguish these 3 phytoplasmas is important because there's going to be differences, to some extent, differences in host range. So we want to be sure which pathogen we're dealing with in a given infection. Now, it appears that Haplaxius crutus is the vector of all 3, but we don't know if there's other insects that can transmit these phytoplasmas. So it's another reason why it's kind of important to know which pathogen we're dealing with, given that it's possible that other insect species can transmit one of the phytoplasmas and potentially not the other. So this is a new test that we're now implementing when you send samples. So on the submission form, you have the 3 options for the qPCR, the duplicate qPCR, which we basically just run it again to be sure, and the digital PCR. But the qPCR test that most people select and we do on a given sample is actually comprised of 3 separate tests. So you're really getting 3 separate tests on a sample, where you have basically quality control and checks that the strengths and weaknesses of each test kind of overlap and cover it, so that when we send the results to you, we're confident in the data, because I know a lot of times a positive means the palm's going to get removed, and the last thing you want is a false positive and remove a plant that's not infected with phytoplasma. So the one test the basic test is a very specific test that pretty much only is going to amplify phytoplasma. It's not going to pick up other bacteria that are present in the environment. And then we have two other tests that help to identify which phytoplasma we're dealing with. One is based off of one gene, and then this new one here is based off a gene called SEC-A. But there are two different genes, and we have those to distinguish which phytoplasma we're dealing with. So if the initial test fails and this test gives us a result, we know that it's not a phytoplasma. It's just ambient bacteria we're detecting with the secondary test. And then the first test, if that comes up positive, then the second two tests that we run, then we use that to distinguish which phytoplasma we're dealing with. Now, up until recently, we were running our digital PCR test that was available primarily for bulk testing. So if a nursery or a landscaper has a large number of plants that they wanted to verify were healthy and free of infection, the digital PCR test was a cost-effective way of doing it, where you could pool, we were marketing it like 20 samples. So if you have 20 palms, you can test them together for $200 to confirm that they're infected. Now, if it is positive, it didn't distinguish which palm or how many were infected, but it was meant to be a cost-effective way to verify everything was healthy. So that test was $200 a sample, simply because the supplies for running that and the labor costs are substantially higher than the qPCR. Now, we have upgraded our system and got a new model for the digital PCR testing that basically the workflow is now comparable to the qPCR tests that we do. So now we were able to reduce the cost of the digital PCR test to $100 per sample. Now, the qPCR test is still $75 per test, but with the new digital PCR test, it's $25 more per reaction, but you get 100 times more sensitivity. So here it's a little more, it's more affordable to test an individual plant using digital PCR. And this is very helpful if you're trying to detect the infection early in a plant. So if a palm looks healthy and you're not sure if it's infected yet, you want to have a higher degree of sensitivity when testing that plant, because if it's just a recent infection on that plant, the titer might be a lot lower. So having that extra sensitivity is going to be critical. Now, if a plant has symptoms, you don't need to do digital PCR. Once there's symptoms, the phytoplasma is fully systemic, and there is nowhere on that plant where there is not phytoplasma. So if you're testing a palm that's symptomatic, stick with the qPCR. The basic test is adequate. But if you're not sure on a plant, if it looks healthy and you want to test it, the digital PCR is going to be a safer test to go with. So sample submission is, if you're not familiar with it, well, we ask for 3 grams of tissue, a film canister's worth, and I'm realizing that as I get older, I need to change my reference for what constitutes 3 grams, because the younger and younger kids don't know what film canisters are, so I'm going to have to find a better reference there. But basically about 3 grams. That way we have some wiggle room if we get an ambiguous result, or if there's a mistake or something happens, we like to have a little extra. And basically the tissue we request is living trunk tissue. So you got to drill past the pseudobark and then get the white living trunk tissue on the inside. These samples can be shipped overnight by FedEx or UPS. If you can avoid using the postal service, not that they're not good on getting the samples, but the postal service samples end up in my mailbox, and the administration at the center, they don't notify me when I have things in my mailbox. They only notify me if a package comes and it's signed for on the cart in the main office. So if it comes in by FedEx or UPS, I get an email. If it comes in by USPS, it sits over my mailbox until I stop being lazy and walk across the parking lot to check it. So I've had a few cases where samples sat longer than I would have liked. So try to use FedEx or UPS, or if you do USPS, just give me a heads up by email and say you got a sample coming in. That way I know to look for it. The instructions and submission forms are available on my website, bahderlab.com, under the services tab. And that's where you'll find all of the submission forms and instructions for sampling and testing. We do have an option for processing sticky traps as well. So if you're interested in looking at insect vector populations, you can set up yellow sticky traps, send them to me. I'll do the counts and look for Crudis. That's absolutely free. And then basically what happens is I'll take your traps, count the number of vector on there, and basically follow up with you and say, okay, you've got a lot of insects. And then determine if you do want to test some of the bugs to see if they're carrying the phytoplasma. And then we offer this and do this because sometimes I get a lot of questions about whether it's safe to replant a palm in an area if it died from lethal bronzing. And, if there is, with that type of data, if we set out sticky traps and for a couple of weeks we're not collecting any Hoplaxius crudis and they're not showing up on the sticky traps, it's an indicator that it's safer and you don't have to put as much care into protecting the plant that you're bringing in, other than making sure it's not already infected. Now, if you send me sticky traps and they're covered with Hoplaxius crudis and we test them and the insects are carrying the phytoplasma, that would indicate that where you want to plant your new plant is a high risk that plant gets infected. So prior to installing it, you're going to want to, hit it with OTC and a heavy dose of insecticide if you're going to continue or pursue planting that palm in that area. So it's useful for that. And it also kind of gives us an idea on the distribution of the vector when it's most abundant. So, everybody wins with that. We get data and you get better insights into how to deal with and protect your palms when you're planting them. So with that, that's where we currently are at with lethal bronzing. And I'm more than happy to take any questions that anybody has.
You got quite a few questions in this chat.
So no doubt.
So I guess just since you're fresh off of that, with the sticky traps, people are asking like what the sources are, and then if there's a specific height that they work at better.
Sure. So, I mean, there's countless varieties of yellow sticky traps on Amazon, Home Depot. I mean, I think you can get them at Home Depot. I haven't looked specifically, but you can order them from Amazon. Any type of yellow sticky trap is going to be perfectly fine. The key to it being useful and successful is it has to be within the canopy. So you can't, if you've got a 60, 70 foot tall palm, you can't put the sticky trap on the trunk. It's not going to do anything for you. So it's going to have to be on palms that are accessible to you, either just from standing on the ground, so shorter palms, or if if a ladder is practical or accessible, that's fine as well. But the straps do need to be enclosed within the canopy.
So someone just asked, follow up with that. Do you need a bait for the trap or anything like that? Or
No, as far as we took some of those volatiles that we found in that study, and we tried putting them on in traps to attract Crudis, and they're not attracted to it. So just the passive collecting of the trap can be effective. We've compared colors of traps, and there are white sticky traps can do a little bit better, but not to a point where I would say you have to use white traps over yellow. Yellow sticky traps are perfectly adequate for population studies. I'm using them myself in a few experiments I have at some of the at the Montgomery Botanical Center. I'm using yellow sticky traps there, and they're perfectly fine. White can be a little bit better, but again, it's not a big deal. But they don't need any kind of extra chemical bait for the traps. Just the sticky trap's going to be enough.
okay. And is there any specific time interval between like retrieving them or any?
Yeah, that's a great question. So if you want to test for phytoplasma, switching them out every week is going to be critical. If you can do it every 3 to 4 days is better, but a week and you should be fine. If you go beyond a week, you start getting significant degradation of the DNA in the samples to where you can still detect it, but it's much less reliable. So if you're going to be testing for phytoplasma, you can that needs to be done every week. And then if you're just interested in assessing if the insect is there to see if the population see what the population is like, you can do it. The maximum you can leave traps out is a month. After that, the bodies start to break down, and it's really hard to identify them. I mean, I can figure them out, but if I have somebody else looking at traps, it gets a little bit harder to visibly ID them. 2 weeks, everything still looks like it did when it was alive. So 2 weeks is optimal, but no more than a month. If you're just visually looking to see if the bug is there, then if it is, then you can start doing a more shorter time period if you want to test for phytoplasma.
Cool. Okay. Let's see. We've got a question that just came in. The EPA has proposals pending to restrict the use of neonicotinoids. Is there another insecticide that is being tested that's known to work?
Yeah, so there are other insecticides that work. The ones that I know are dimethoate and diazynone. Now, I don't recall if those are neonicotinoids or not. And unfortunately, some of the new compounds that have been released were developed and are proprietary from Syngenta, and we were doing experiments with that, but that's been shut down because of the business and politics behind Syngenta. So that's right now, imidacloprid works. So, stock up. But basically, that's one of the reasons why I'm so eager to look at this volatile as a because it's a natural compound the plants are producing. So if we can harness that to manage the disease and in the plants, that's a truly green solution that that's also cheap, easy to produce, and easy to do large scale. So definitely preferable over insecticides. So that's kind of where we're headed.
Okay, I guess got a lot of questions here. So I guess going back to 3-hexanol, do all palm species produce 3-hexanol? And also, like, a follow up to that, is 3-hexanol found in any products by chance?
So those volatiles that I showed are they're basically ubiquitous in plants. There was a study I saw a news headline for recently that was kind of funny, that said when you're cut your grass, that fresh grass smell when you cut it is basically the plant screaming for help. And basically these different volatiles that we found in the palms, they're not unique to palms or to lethal bronzing infection. They're released when it's a significant stress. And actually, I'm kind of curious if some of these compounds aren't just effective against lethal bronzing, but if they could help in, against potentially fungal infections or other things like that. So yeah, 3-hexanol is found in all plants as a defense response, and whether or not it's in products available now, I don't know that.
Do you have a recommended frequency for spraying, like spraying regimes?
As far as I'm aware, the label rate and persistence, if you're going with imidacloprid, is like every 6 months, is what's recommended.
Okay. Okay. Stuff related to grass, and controlling the different phases of crudis. How short to mow the grass in landscape and nurseries, and what distance to remove grass from the palms?
So I don't think the length of the grass is a critical variable. I think it's more important the moisture availability. So, like, for example, in the palm beds where it's kind of raised and drier, the grass is extremely short. But there's no water up there, except for when they irrigate or it rains and it gets a little bit of moisture. But basically, water runs off from those raised beds, and it's significantly drier than the ditches. So I think the moisture availability is more critical to the nymph preference and the breeding habitat. Now, because I have seen in like really like standard St. Augustine grass that's mowed normally, you can find nymphs of crudis. And not that this is a great example, but I have noticed around in like my yard, for example, I've seen the nymphs of crudis crawling around the base of the sprinkler heads. So where there's, water that kind of seeps out or drips, and it's a little damper than the rest of the yard, you'll tend to find the nymphs crawling around the sprinkler heads, and also around shorter palms. We've found the nymphs at the drip line. So where the fronds end, and when you get irrigation or rain that just drips and make that like ring of moisture around the plant, you tend to find more nymphs in that part. So in the nurseries, it's fairly straightforward to rototill and remove all the grass from the ditches, and that'll be effective. And you can't do that in the urban setting unless you can convince people to remove all their grass. But I would say if you were going to do that, I would remove the grass and mulch the palms maybe a little bit past the drip line of the plant, and that may go a long way towards lowering the vector population without insecticides. But again, we haven't done that work to be sure, but it's something that is could be a preferred alternative to insecticides, is just mulching around the palm. Now they could still be breeding in other habitats, but they don't like to go far from the host plant to lay eggs. So now that may or may not be an option in all areas. So
I guess just a follow up to that, is there anything related to like turf species? I mean, I guess it would probably be really related to like whatever retains moisture the most would be my guess. But have you guys looked at any of that, or?
they definitely do have preferences on which grasses there are, but so in the nurseries, something like guinea grass, or there's another species where you have like a common cluster where all the grass kind of arises from one point, and you get this kind of almost like a bowl, and it's like this thick area of the leaf bases. They really like those types of grasses, and they prefer those over things like St. Augustine grass. But in urban settings where it's pretty much just St. Augustine grass, they'll go to that without any hesitation. So
Cool. Well, I want to be mindful of your time. Like, if you've got another 10 minutes, I'm sure we could keep passing through. Okay.
sure, yeah.
Has there been any work with like also injecting imidacloprid in addition to oxytetracycline?
I've heard of companies doing that, and I think they were focusing on white flies, and I know they inject the two together, and from what I've been told, when they inject imidacloprid into the palms with white flies, it doesn't take long for it to take effect and start killing off the white fly adults. And if it's doing that quickly, the timing should be similar in Hyplaxius crudis, because crudis and whitefly are feeding on the exact same tissue type. So they're both tapping into the phloem in the vascular network. So if somebody's treating palms already for like whitefly or something like that, I mean, they don't need to do anything extra for crudis. They're already treating it. So
Has there been any luck with another insecticide mode of action? Merit is overused now in the landscape because of the ficus whitefly management.
yeah. Like I said, there's hasn't been too much work on insecticides, because in the past, the research on this disease and insect were very basic. They weren't really looking at, they weren't doing a lot of applied research. And now that we know what the vector is and starting to learn the biology, we're starting to explore these different options. Now, unfortunately, we've hit this point where because palms are a primarily Florida issue and they're not a food crop, there's very little interest in funding it. So we're kind of at a difficult point where a lot of the agencies that have funded it, I'm starting to see LB fatigue, at least in terms of people's pocketbooks. But hopefully, if we can get some support from USDA or something, we can start exploring other insecticides.
Is it Are you curious? I guess just following up, like was someone put in the chat here. Are you interested at all in doing stuff with growth regulators?
Um, yeah, I don't see that being an issue, and it would be interesting to do. The nice thing with the nymphs being in the grasses is that it's an easy target. It's easy access. You don't have to get up into the canopy of the palm or anything like that. Again, the challenge is knowing the distribution of the nymphs in the urban setting. Basically, at this point, I'll kind of revisit the nursery scenario where I really wouldn't recommend anything other than rototilling those ditches once a year. That's going to be do wonders towards reducing your vector population and ultimately the disease pressure. In the urban setting, it's a little trickier, where we don't know what the vector distribution is, and it's not going to be the same from place to place. So what you find on the median is going to be different from a park, from somebody's backyard. So we would need to really understand what type of breeding habitats were preferred in these different areas and urban settings before we did anything with looking at these insecticides. Now, the imidacloprid was ideal because it's already in use for turf grass for other stuff, and it's already going down. So, it wouldn't really require anything extra other than just treating your turf grass with how it's been done. But as far as exploring new insecticide, just the support hasn't been there for doing that. So
I'm going to try and conjoin a couple questions here. Do you expect like the populations to show like cyclical cycles with Crudis? And then I guess with that is on top of that, maybe somewhat related, is there an optimal treatment window as well, like within the year?
Sure, that's a good question. So whether or not populations are cyclical over the long term, we don't know. I'm currently monitoring populations to see what it does over time. But in general, every year, like there's always adults around, but they definitely peak in like August and September. That's when you get your peak population. So if you do want to do a more surgical or precise treatment with an insecticide, so let's say you could treat in like May or June to target that August population and wipe out and hit them hard when they're going to be at their peak. Then you might be able to get away with not treating twice a year or every 6 months and time it so you're just going after that peak abundance of the adults in the palm canopies. Now, the benefit, and that we have confidence in because we found that usually the vector population increases in August, September, and then in the spring of the next year is when you start to see a lot of the new infections show up. So and that's happened over 6 years, and an independent lab has similar data. So what happens is if you can knock that vector population back in the August, September period, that may go a long way to reducing the number of new cases that start to show up in the spring. So yeah, I would say August, September is when it peaks. So if you can get something down like in May or June or July so that it has time to become fully systemic in the palm, then that's going to be go a long way.
Okay, cool. I've got some questions, I guess, about like species. I think that you've got a pretty extensive list of like palm species that are impacted. Um, I guess someone was asking specifically about foxtail, but I'm gonna throw on top of that, like, um, I guess, are there any species I think we typically think of Sylvester's as like the main thing that is susceptible to it. Are there any other? Is there been any further correlations or data that's come out there showing like what shows the highest mortality if affected?
Yeah, so from the clinic data, it's kind of hard because there's, you know, Phoenix palms and cabbage palms are the predominant plant ornamentals in Central Florida and very, very common in South Florida. So those are the ones we get the most infections from, but is that because that's just what everybody's planting or so on? Now, from much to the annoyance of my center director, we've been letting the disease go on our research center because unfortunately, sometimes the best way to learn about a disease is to just let it go and watch it. And in this context, we have a variety of, we have Phoenix palms, cabbage palm, coconut, Christmas palms, foxtails, all the standard ornamentals. And under natural conditions, it seems that cabbage palm is the most susceptible. Now, one of the points to this is, and we now that we know what the vector is and we can control infections and do transmission experiments, the susceptibility versus resistance, right now we can't say with confidence if it's true susceptibility or if it's vector feeding preference, because in this natural setting, these insects have access to all the different palms and there's no management going on. And pretty much the only plants that are dying are the cabbage palms and some coconut, and sporadically Phoenix robellini, Christmas palms. And that, and given that so many Phoenix palms are dying in like urban settings, sometimes that's the only species the insect has access to. Whereas on the center, it's kind of more natural. So we get more cabbage palms, but I don't know if that's because the vector prefers feeding on it or if it is truly more susceptible, or excuse me, yeah, it's susceptible. So eventually we'll be able to do the experiments where we expose all these different palm species to infective insects and say, yes, royal palms, foxtails, and Bismarcks are resistant to the infection. And then our controls would be cabbage palm and Phoenix palms to say, basically, those are our positive controls. We know they're infected, they're susceptible. And then if we expose these other palms to the infection and they don't die, then we can say, okay, they're resistant.
Cool. That's much more complex than I thought it was going to be, like in terms of
yeah, because there's definitely a feeding preference for Crudis. I've noticed more, but again, this could be not the palm preference itself, but more the habitat for breeding. So, Crudis may not feed on a Phoenix palm, in the middle of the city simply because there's nowhere for it to lay its egg. So the environment can influence the population, and then that ultimately influences the feeding preference. So it's complicated for sure.
Well, cool. I think that's probably a good point to end on too. I guess you'll be at more training events as well, and like with ISA and
yeah, I should be. I know, we're doing Palm School. I think we've got one coming up in Jacksonville in October. So And eventually I'm thinking of doing a workshop to teach people how to identify the vector. So how to sample, put up sticky traps, to identify it through like a hand lens, so on and so forth. So that may be something in the near future to look forward to.
Well, let us know if you need some help. That sounds really cool.
Okay.
So, yeah, I guess we'll follow up with everybody. If you got the tickets to get CEUs, we'll shoot you emails to get and make sure that we get like your information, like and we'll follow up with the forms as well, and we'll give you guys all recordings. Thanks for being here. This was a really good webinar. Thank you, Dr. Bahder.
My pleasure. Thanks,
Well,
everybody.
yeah, I feel like this is I've learned a lot about I mean Crutis and LBD in general. So but yeah, I guess everybody take care, and I guess I'll follow up with the post information and resources afterward. So take care.